So, if you take one language and you move it into another language where it still makes the same sort of sense, that's translating. So they decided that that's what the second thing in this sequence was going to be. You're taking a nucleic acid sequence and changing it into an amino acid sequence. Two very different types of molecules, like two languages. And so we talked about transcription, taking the code from the DNA and converting it to messenger RNA. And now we're going to talk about the translation, taking the code from the RNA and translating it into a sequence of different amino acids. This one, you're going to have to write along with me because I got pictures. So here's your messenger RNA, mRNA, coming out of the nucleus, going wherever it needs to go. It could be going through the cytoplasm, could be going through the endoplasmic reticulum and attaching to a ribosome embedded in the endoplasmic, the rough endoplasmic reticulum. That's a very common destination. But very often the ribosome is kind of shown as kind of an acorn-shaped structure where the rim of the acorn is where stuff happens. This one is a different picture, but the same basic idea happens. As this feeds through, you have these little molecules called transfer, little t, RNA. And each transfer RNA has a 3 amino acid sequence on this side. Here's your codons in your RNA. Here's your anticodon in your transfer RNA. And so it will match up with a particular sequence of 3 here. For every anticodon, there's an amino acid. So the transfer RNA that hooks on here will be bringing in a particular amino acid. So that's how this code gets translated into a string of amino acids. As this feeds through, it brings the amino acids close to the next one that's attaching. This connects here, this releases here, and this comes off to grab another of the same amino acid and be available to come back in. Just to give you some idea, there's your anticodon. This is what holds on to the amino acid. This is the structure of it. Here's your anticodon down here. Here's your attachment site, little bitty attachment site here. Another way of showing it, I think that shows what's happening. This looks like an acorn. It shows what's happening here a little bit more clearly. Just read the past week, essentially the failure of a what's called an antisense treatment. Some genetic diseases come from bad alleles making not just non-working proteins, but proteins that do bad things. And one of the potential ways to block that is to come up with a molecule that will attach to the messenger RNA when it comes out and keep it from being translated, antisense. And they were trying to do that with Huntington's disease, and it turns out not to have really particularly done a good thing. It's very difficult to get to work, probably because it's hard to get those molecules into anything, but also they wind up attaching to things you didn't expect them to attach to and doing bad things beyond the good thing you're trying to get them to do. It was going to be the next big thing in pharmaceuticals, I want to say 20 years ago. And there's a couple that work, but for the most part, they don't. So here's your messenger RNA feeding through. Here's one transfer RNA. Here's another one coming in to attach. Here's them adding to the string of amino acids. There it is, and being added to. Now you've got a new string, it moves into the next spot, and another one will come in and attach. And there they've got the stop codon marked. So that you didn't stop making the messenger RNA at the stop codon, you made, you sort of threw a tail onto it, because this is showing the tail. But that's not going to get translated, it's going to be translated as a let go. Let me see. Oh, here's a prettier one. You can expect this in one of the questions. There's only about 21 amino acids that are typically used to make proteins. And even then, one of them very rarely winds up in there. There's a lot more than that if you're looking at 3 nucleotide sequences. So you've got a 4 by 4, well actually a 16 by 16 block here. And this is a way to translate the codon sequence. Oh, I need to tap here, not there. Into the amino acid sequence, where if say the codon sequence is AAA, there's your first A, so it reads across here. What's the second A? Here's your second A, it reads down here, so now you're just in this box, and here's AAA, and that translates to lysine. If it was CCC, here's C reading across, here's C reading down, here's CCC in the box. If it was UUU, just to keep it simple, U, 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 U. And any other sequence can be here. Here's your methionine or start. There's 3 different stop codons. Now notice leucine, proline, serine, leucine, that the same code, 6 codes will give you leucine. 4 codes will give you proline. 4 codes and a couple over here will give you serine. So this is why a lot of point mutations don't change anything, because you can change one of these codons into a similar codon and get the same amino acid, or get one that's very similar. These are very similar amino acids in terms of how they connect to each other. So if you change an amino acid into one that connects along the string pretty similarly, it's not going to change the protein. So point mutations don't tend to change much unless you change a whole bunch of codons in a sequence, which we'll talk about. This is called genetic redundancy. The fact that several different codons will give you the same amino acid. It turns out, again, in actuality, if you change the codons, it may not, it may affect the reading speed or the translating speed in the ribosome. So your ability to make something at the same rate that you need to make it may be affected by, even though it's the same amino acid, it may not come off the string quickly enough. Speaking of coming off the string, if you were to make this in a test tube, put the amino acids next to one another, most of the time there are various ways that the amino string can kink and curl and cross-attach, and so you're not always going to get the same secondary and tertiary shape, the structure. You're not always going to get a protein shaped the way that you want the protein to be shaped, which makes sense because a lot of times the proteins are shaped a certain way to keep them inhibited until you want them working. Or you want to be able to inhibit them later, which involves changing the shape of the protein without changing the amino acid sequence. As these things come out, there are a collection of proteins waiting for them that make sure that they form the right configuration, they form the right tertiary structure, or grabbing them and fitting them with another one if there's quaternary structure. Those molecules that make sure they get formed properly, they get the right shape, are called chaperonins. Somebody goes, yeah, it's like chaperones, making sure everything happens right at a dance. Sometimes when you look at stuff in Latin, it has kind of the same sort of silly roots, but when it's English and you speak English, you go, okay, really? One interesting thing about chaperonins is that that class of proteins was already known when somebody started to figure out what the chaperonins were, and somebody went, okay, I know what those are. Those are heat shock proteins. And in prokaryotes that live in extremely hot environments, what they call extremophiles or thermophiles, is that the heat denatures the shape of the protein a lot, and there are these heat shock proteins to put it back into the right shape so it can keep working. As the fluctuations in heat have an effect on the proteins. And so it's doing kind of the same thing at a different stage in the issue. But the idea of proteins that keep another protein in the right shape or help another protein go back to the right shape, shouldn't have been a huge surprise that they're the same basic type of proteins. There also are going to be additions later on, both quaternary structure and what are called prosthetic groups. Groups that are non-protein groups that are added. It may be minerals, like if you're going to, or mineral groups like a heme group going into hemoglobin. But your functional proteins, remember, have pieces to them, domains, that do different things. And some of the domains are not necessarily amino acid sequences, but they need to be added to the protein for the protein to actually be functional. It's true for chlorophyll as well. So hemoglobin, chlorophyll, a couple of really important molecules need prosthetic groups attached before they're actually ready to work. Okay, that's a good place to stop. Here's a fairly short video.